He's a man from down under, but one wonders if he blundered by heading north to where the sun doesn't shine. He crossed the line, the 49. Oh, Canada. It takes stamina to live in the dark and the cold, but then again, Stuart is bold. He's the steward of pines and maples growing, great prairies spreading, and lordly rivers flowing, cold, wet winds ever blowing, science showing. But before he was bestowing Vancouver with his genius, before the frost set in, he was traveling the world so often. Prior to Canada, he came from Boston. Prior to that, he did his PhD at Oxford. And before that, he schooled in Sydney. His research is not in the kidney. Do you know how hard it is to rhyme the word Sydney? He studies immune deficiency and he thinks fast, susceptibilities to infection. Hey, these kids aren't going to last. He studies allergies and genetics too, and gets deep into the shit with the microbiome. Ew. Immunology today is precision health, and Dr. Turvey today will offer a great wealth of knowledge. And so I hope you'll join me in welcoming him to the UC. Okay, stuff I couldn't work into the rhyme. I've known Stuart for about 19 years. We both were at Boston Children's together, and he is the Tier 1 Canada Research Chair in Precision Health and is the Aubrey Tingle Professor of Pediatric Immunology at the University of British Columbia. He's a pediatric immunologist who works at BC Children's Hospital and also directs child and family research there. Welcome.
Okay.
We can break this.
We're gonna share. Just share. Is that sharing? That's good.
Yes, that's sharing.
Okay. Well, thank you for that introduction. It's tough for me to beat that and to offer some rhymes. We gotta share.
Do you want to? Yeah. Sorry about that.
No. Great.
There we go.
So I'm delighted to be here, and my goal today is to talk to you about, I gave it the title, The Power of Partnerships, and it's really, I think, an example of partnerships where clinicians identify patients and clinicians work with scientists, and we work with scientists across the world to advance knowledge and to begin to define new human diseases. So it might seem a little corny, but I think there's a really nice metaphor here about working together in this story, and about how that can advance global knowledge. And I want to say that rather than sort of having this as a long didactic session, I'm happy to have a conversation. So if people want to jump in and ask questions on the Zoom or in person as we move through, happy to clarify things as we go along. Let me just get the pointer going. There we go. So this is me. I'm a pediatrician. So everything I do is really embedded within the questions that we see every day in the clinic. And here I'll be talking about stories that evolve from patients we saw in the clinic who didn't have a diagnosis, didn't have an obvious answer for their many clinical problems. And I sort of find this inspiring and think we can, all of us who might work in the clinical realm, realize that there will be important patients out there that are coming to us that can help us understand new forms of biology. And really, I think at a big level, what we're talking about here are rare diseases, and this would be nothing new for this audience, but rare diseases really matter in pediatric medicine. So it's estimated that something like 1 in 3 children admitted to a pediatric hospital have some form of genetic rare disease. We're making good progress through many global initiatives, but still many have no known cause. And the real tragedy of rare diseases is the concept of the diagnostic odyssey, where patients and their families see specialist after specialist and go through painful after more painful tests, trying to find a diagnosis where they don't get one. And the hope and the opportunity that we have now with precision medicine is that we can start to understand the molecular underpinnings of our patient's disease and then use that information to make a diagnosis, potentially to offer therapy, and to avoid complications that we may be able to predict more accurately. And this is an older slide now, but I think emphasizes that we're really in a golden age for discovery of single gene defects, Mendelian defects. So in 2010, we had the first application of whole exome sequencing. So the first broadly applied form of next generation sequencing. And then since then, this next generation sequencing, initially exome and then more commonly whole genome sequencing, has empowered the rapid diagnosis of more and more patients. And I'm a clinical immunologist, and so it's equally exciting times for us. And so this paper just published suggests that we're close to 500 single gene defects that affect the immune system. Manish suggested that we trained together here back in 2003, 2004, 2005. And, we were in Boston and we were arrogant trainees who thought we knew things. We only had to know 100 diseases. So any trainees who are in the space now, it's much harder. And I can promise you that Manish might know the 500 single gene defects. I do not know them. But I think this is remarkably important. The other thing that's important in immunology space has been our ability to actually make diagnoses in single patients. And in general, in genetics, we're looking to gather multiple patients with the same phenotype and same genotype to describe a new syndrome. In immunology, we have the ability to access the organ. We can access the blood, and we can prove in patient primary cells that this is the genetic defect causing disease. So we shouldn't be afraid of looking hard at single patients and working up those single patients, because that can truly empower discovery. And so the diseases we're talking about, we used to call them primary immune deficiencies. Now we're really calling them inborn errors of immunity. And these are genetically determined disorders of the immune system. They greatly enhance our susceptibility to infections, but also autoimmunity and malignancy. And they're not so rare. And I think, although individually these conditions are rare, in aggregate, they have prevalence, some estimates as common as 1 in 500 to something like 1 in 10,000. So it means that these are out there in a decent sized practice of many physicians. Our ability to make that diagnosis can be life-saving. With the knowledge, we can provide accurate genetic counseling for the affected individuals. And I think really most relevant for this group is the idea that these patients with profound single gene defects provide important insights into fundamental human biology. And so as the field of inborn errors of immunity grew up, there was initially a very heavy focus on the infections. And so here we have the 10 warning signs published by the Jeffrey Modell Foundation. You can look through them. They're talking about too many infections and identifying people with too many infections. On the right, we have the bubble boy living in his bubble, being protected from infections through that technology. But we've come to realize that while the infections had been really at the forefront of our understanding these defects, we now realize that malignancy, autoimmunity, and allergy are also a clear factor that occurs in patients who have just a dysregulated immune system. And so one of the things that our lab has been focusing on has been these what are called primary atopic disorders. So this was a term coined by John Lyons and Josh Milner back in 2018 that was really looking to emphasize single gene defects that present as allergic disease. And they recognize that there's now probably around 40 inborn errors of immunity that could be characterized as primary atopic disorders, and they affect and impact many different mechanisms of immune function and immune structure. And on the right here are some of the sort of classic disorders that we might recognize as primary atopic disorders. And so our lab has been interested in this, and we've been bringing together, trying to sort of raise awareness of these and identifying the clinical red flags that might signal that a patient has a monogenic allergic disease. And not surprisingly, they had atopy, they had allergic disease, they had high levels of eosinophils and IgE in their blood. But when we reviewed the literature, they also had other features that were perhaps less expected. So there was malignancy, much more common, the growth failure and short stature, connective tissue disorders. And so we've started to sort of paint a picture of these patients with primary atopic disorders, looking to raise awareness, because truthfully, most of our allergy colleagues are thinking of their allergic disease that they study as a polygenic disease. And for the vast majority of patients, it is. But for some, there will be a single gene defect, and it's life-changing and transformative if we can identify that defect. And so I sort of told you that this was about partnerships. And so I was preaching this and encouraging my clinical friends to be thinking about single gene defects as a cause of allergy. And a senior physician came over and wanted to tell me a story about a patient that he'd seen many years ago. And, you know, that sounds like anecdote, but interestingly, in the rare disease literature, there's actually quite strong evidence that a very good predictive factor for having a single gene defect is the need to see multiple specialists and multiple experienced specialists saying there's something wrong here. I don't know what it is, but there's something wrong. And so I tend to take these conversations seriously, and particularly this clinician who'd identified with us several other single gene defects. And at the risk of making this sound a bit like a clinical grand rounds, but this is the main slide, I just want to tell you about the features of this first patient that he came to talk to me about. So he said that this was a patient that he'd followed at our children's hospital as a child about 15 years ago. He was the only child born to unrelated parents who really were healthy. But this baby was sick from very early on. The first thing was severe widespread atopic dermatitis or eczema that developed at 1 month of age and was resistant to most known therapies. We tried therapy after therapy and never got the skin under control. The other huge feature was issues with the gastrointestinal tract, where there was eosinophilic infiltration and eosinophilic gastrointestinal disease. And so that meant that the baby had reflux, so was vomiting, needed a surgical procedure called a Nissen fundoplication, where they really try and tighten up the esophagus so that the things don't reflux back. And in fact, around age 1, had to have a gastrostomy tube placed. And so for his life, was predominantly fed through this tube, hydrolyzed or elemental formula. And his ability to eat by mouth was limited to essentially potatoes. He had multiple food and drug allergies and had episodes of anaphylaxis. He'd been admitted with asthma. He didn't grow well. He didn't have so many infections, which is sort of what we always ask about. He had the infections that we might expect with disrupted skin. So infection of the skin with Staphylococcus aureus, strep group A streptococci, Candida, and then virus that causes a condition called molluscum contagiosum. The physicians across the years had tried many treatments. The list is long. And really the only thing that helped control his disease in any way was oral dosing of steroids. And so he needed decent doses of corticosteroids every single day. So the dose itself doesn't matter, but just to know that this was a solid dose received every day that then caused many side effects that we'd expect with that. And his blood testing really just showed high eosinophil counts in the blood and high serum IgE. But the other tests that came along over time gave no answers. And I'll just give a postscript here that this patient actually died of a cerebral hemorrhage at age 35, so sort of after this. And that too may be part of the phenotype we worked out based on this patient. So I've painted you a story of a patient who was very sick from early on in his life. Here's the growth chart. You may not be familiar with these. We pediatricians love to plot the height and weight of children as a very good integrator of overall health. And what you can see here, for example, in the height, is that he often was below the 3rd percentile, and similarly for weight. So this would meet criteria for failure to thrive, growth failure, waxed and waned over time, but this was his growth chart. So our approach to this, I think, is the same for hundreds, thousands of labs around the world looking at this. So we work with the clinicians and enroll the patient and the family in a research protocol. We'll apply some form of next generation sequencing. Here we performed whole exome sequencing on the trio of the healthy parents and this patient. As time gone on, like everyone else, we've expanded to whole genome sequencing. After bioinformatic filtering, we'll find compelling variants, and if we're convinced, we'll take it to the lab to start to understand if this variant might be the cause of the patient's disease. My passion is the idea that with that knowledge, we can start to develop therapies or repurpose medicines that are approved that might help reverse or control their condition. And then, we'll move on a process to look to identify, once we're convinced that this seems to be real, we'll look to identify other similarly affected patients. And so for this, we used a matching service called Gene Matcher, where we can post our gene, and if others post the same gene, email links us, and we can talk about phenotypes. And so we did that, and we're actually matched up with someone to call me. So we were matched up with actually another Canadian patient in Ontario who had a genetic change, and we worked with that patient. Then we had 2, and we said, well, maybe we should look. There must be more. So we posted a preprint of this story on medRxiv, and then very quickly we were inundated with what seemed to be a huge number of these patients. And so I think through this iterative process, what we learned, and what I'll try to convince you of, is that we've identified a new human condition caused by germline heterozygous gain-of-function variants in the gene STAT6 that underlie severe allergic disease. And so let me take you through that story and take you through that data. So before I get into that, is everyone happy with introduction? No? Okay. You happy? I'll keep going then. I'll try and rhyme next maybe. So let's talk about STAT6. So people might know about STAT6, but others mightn't. STAT6 really is a transcription factor that lies at the heart of allergic inflammation. I, as a PhD student in Oxford, learned that if you had STAT6 knockout mice, they couldn't get allergies. STAT6 was a kind of obvious gene that we in the clinical immunology field thought there must be some changes in patients with STAT6 related to allergic disease. So STAT6 is a transcription factor that's key for transducing the signal from the classic allergic cytokines, IL-4 and IL-13. When they bind their appropriate receptor, they activate the JAKs, and then the JAKs phosphorylate themselves and phosphorylate STAT6, allowing it to migrate to the nucleus, where it can drive expression of pro-allergic genes. is looking at that another way here in the context of asthma really, is that IL-4 and IL-13 kind of sit at the center of the allergic universe, and their signaling through STAT6 is a key driver of allergic inflammation. And although we hadn't identified patients with single gene defects related to STAT6, there'd been decade of genome-wide association studies that had linked common variants in STAT6 to human allergic disease, and the list is long and links to essentially all the measures of allergic disease that we might think about. So STAT6 was an obvious and very compelling variant to explain these patients' phenotype. And so I told you we started with one patient, and then through GeneMatcher we had another, and then through medRxiv and writing email to kind of everyone I knew, we rapidly expanded our cohort to 16 patients from 10 families across three continents. And I'm sort of proud of this map in that I think it actually reflects the culture of the global clinical immunology community and the willingness to partner and work together to define our patient diseases. So this was a huge pleasure to bring these people together. And in fact, it was really easy. People really wanted to work with us and to advocate, for their patients to help them get a diagnosis. And so I'm, proud to be part of, I think, this very supportive global community. And here are the pedigrees of the patients. I realize it's small, but I think that the messages can come through. So we had 10 families with 16 patients. And when we start to look at this, you can see some different patterns coming through. So some, there was an inherited pattern, for example, this family, or this family, although I'd emphasize this patient seemed to be affected clinically, but we were unable to sequence them. And we see that that's affecting offspring of different sexes, and so is consistent with an autosomal dominant inheritance pattern. But in addition to that, we had clear sporadic disease, where unaffected parents would have a single affected affected child. And so in most of these, and again, the reality of human studies is we couldn't always get every single family member to sequence, but in most of these, in the sporadic cases, we were able to establish that these were de novo variants, so not detectable within the blood of the parents. And so I think this is sort of interesting in that we're starting to, with this condition, explain autosomal dominant family patterns. These are the ones we'd be aware of. We always ask about family history, but many of them were de novo. So brand new de novo variants appearing in the child. So this is the inheritance pattern you had of these various families. And I won't belabor the point, except to say that these patients clinically really all look like that first patient I told you about. They all had early onset atopic dermatitis. They had the food allergies. They had the eosinophilic gastrointestinal disease. They had a persistently high eosinophils and IgE in their bloodstream. And then 2 of the patients in the cohort died related to their, I think, likely related to the condition. I think the anaphylaxis was almost certainly related to the condition, and the cerebral hemorrhage from an aneurysm may well be related to the condition, in that variants in other stat molecules are associated with changes in the CNS vasculature. But again, it's really going to be more patients being recognized that allow us to truly define the phenotype here. I'll take you through some photos just to, I think, kind of emphasize this. So here's the skin of one of the patients. This is very severe, lichenified atopic dermatitis. It's very widespread. Biopsies showed atopic dermatitis with eosinophils. This gastrointestinal disease, particularly eosinophilic esophagitis, was common. And the patients had these classic features that the gastroenterologists recognize, with linear furrows and these horizontal rings that are called trachealization of the esophagus. And again, eosinophils scattered throughout on the biopsies. Here's the blood values. We put this on a log scale. So this is the IgE, and here's the eosinophil count. Normal range is shown in the gray. And for the eosinophil count, this dotted line represents a value of 1.5 or 1500, depending on the way you look at it, which tends to be the definition of hypereosinophilia. And so we see patients across the ages at very high serum IgE levels, and also had high eosinophil counts, although we see that they did drop with some treatments over time. This one's deliberately tiny, but this is all the other clinical tests that we do, which are all relatively normal. So looking at blood counts, looking at T cell numbers and T cell subsets, B cells, and so on. And really, there's not much message here, except the platelets tended to be elevated, the white count tended to be elevated, and the serum immunoglobulins tended to be elevated, which is really just a sign of constant inflammation, chronic inflammation. So there was no, I think, clinically obvious biomarkers that would help us identify these patients through the tools that we can access clinically. So let's talk about the variants. Let's dig into them a little. So this is the population genetics. So we're showing the patient variants here in red, and then the data from NOMAD emphasizing missense, and then also predicted loss of function variants. So patient variants were not present in the population databases such as NOMAD, and they had relatively high CAD scores and other in silico predictors suggesting they were likely to be damaging. And this is the variants. So we're not talking about one single variant here for these patients, but they had a number of different variants. The most common variant was at amino acid position 419, where we had a number of variants there. We had another variant in the DNA binding domain, but others scattered a little further, linker, SH2, and then also over here, which is an interesting spot. But when we looked at the 3D structure and we looked at how the STAT6 protein interfaces with the DNA, I think some interesting patterns emerged where these variants were close to the protein DNA interface. Not always key residues, but were close. And this variant out here is unusual and a little interesting in that it lies very close to the phosphorylation site that's key for phosphorylation and activation of STAT6. And so I would say we don't know how, as of today, we don't understand the nuance of how these individual variants are absolutely causing disease, and I think that will emerge with time. But they made sense, I think, in terms of their location where these variants were found. I should have also said with the pedigree is that the disease was 100% penetrant. There was no unaffected person who carried the variants. So it was completely penetrant condition. Everyone who had one of these STAT6 variants had significant allergic disease. And I think this piece is very interesting, and I think this is becoming a recurring theme in immunology, is that what these patients had in their germline were variants that are now known to be somatically mutated in lymphoma. And so this is from the COSMIC database, the catalog of somatic mutations in cancer, which is looking at the somatic mutation count across the STAT6 gene. This is the data from COSMIC. And then we've just sort of highlighted in red where our patients. So this 419 is a hotspot for somatic mutation in cancer, and that the main cancer is lymphoma. So B-cell lymphoma, a massive hotspot for somatic mutations, but also the main position for our patients that we identified, 9 of 16 had a variant at 419. Other variants that we saw had been seen in the COSMIC databases. And I would point out that this family has an affected mother and two affected sons. She was diagnosed with follicular lymphoma at age 40, and then, no, 50, let me get this right, at age 50. And then after treatment, she relapsed at age 60 with a more aggressive form of lymphoma. So activated B-cell lymphoma, DLBCL, diffuse large B-cell lymphoma. So I think, again, this is one patient here, but I think as we look at these patients and study them, I think patients carrying the activating STAT6 variants also likely have an increased risk of lymphoma. And this is the oldest patient in the cohort. So, and she's the one who had the lymphoma, but time, I think, will tell us that. So let's get into the variants a little and try and understand what they were doing. So we started with some relatively simple experiments to start to read out what was going on. And so first of all, we used a classic luciferase assay where we had cells that expressed a STAT6 promoter that would drive luciferase production. And then we used Ranella transfection as a control. And let's go through the data. So we transfected with the wild type and then stimulated with IL-4. And we see a nice signal. We had a number of different controls. This control here is where we mutated the phosphorylation site, so to prevent activation. And again, there was no signal there. And then we used a number of population controls. So controls that were missense variants that were in the scored high on the CAD score, that were in the population genetics, and we included them, and they all performed in a very similar fashion to the wild type variant. And then when we put the patient variants in, we can see they all had a signal, an increased signal at baseline with some increase. Again, this is a log scale, and this system is a little bit saturated, I think, so we're not seeing huge increases with the IL-4. But in general, we'll see that these are all more active. I think one thing to point out is there's a lot of nuance and a lot of difference across the variants in terms of their activity here. We did this with some other luciferase with some other promoters, and again saw a very similar signal where we saw the increased signal, and again nuance based on the different variants, and that's I think an important theme that will come through. We also wanted to look more directly at the activation, and we did that by looking at STAT6 phosphoflow. And again, just using simple flow cytometry, looking at the STAT6 phosphoflow before and after IL-4 stimulation. And again, in the red group here, we see there's a strong signal at baseline that increases significantly with stimulation across those variants. The one big outlier here is actually that variant I mentioned that lies next to the phosphorylation site. We got no signal at all there. We wondered if that might have been something about the steric hindrance, something about changing the structure there would stop the antibody binding. And so to try and address that, we actually did Western blotting. to look at the phosphorylation of STAT6 and compared it with one of the 419 variants. And you can see that there is a quite strong signal with this one, again, suggesting that there's increased phosphostat6 activation with this variant. And so we've started to look at these different variants and understand what the differences are. And I won't get into all the details there, except to just show you this, which is transduction experiments where we put in either wild type or two different variants that were relatively common in our group. And what you start to see is that the transcriptome has some similarities, but has some significant differences based on the patient variant. And I think what we need to start to understand is how this relates to the biology and how this starts to relate to their disease manifestations as well. And so, you know, the way I see this now is really like this is my attempt to have a thermostat. I don't know if it really works, but the patients, you know, have a gain of function in this key transcription factor STAT6, and it's turned up to different extents based on the different variants. And we've seen this, for example, with CARD11, which is, I think, a similar story where there's been gain of function identified and loss of function identified in CARD11. And so with time, we'll start to understand how population variation in these genes changes the thermostat, changes the activity of the STAT6. And so I think that's a work in progress that will evolve as we identify more and more patients. So showing you a lot of experiments in cell lines, and that was feasible in that we could do that in one place and compare the performance of these variants head to head. It was very important to us that we could also show this in primary patient cells. And so our global cohort was a strength and a challenge in that obtaining primary patient cells from Hanoi or Tehran, getting them shipped to one place to analyze them has not been super easy. So I think a weakness in what we have so far is that our data in the patient primary cells is somewhat limited, but we do have, I think, compatible and compelling data. And so one of the things we did first with patient cells was look at the phosphorylation. And, you know, we were excited that the patient cells must show more phosphostat6 and we'll see these really nice differences. And we did that experiment. We did it on these precious blood samples, and there was nothing. And it was like, gosh, this is kind of depressing. Are we wrong here? Do we not understand this? And so we were talking to our big consortium, and John O'Shea from the NIH said, you know, you should look at dephosphorylation. Just as important as turning on the signal is turning it off. And you may start to see some important points emerge in the off phase of this immune response. And he was absolutely right. And here I'm showing it for this one patient that we've done several times, but it's turned out to be true for others, is that after stimulation, the percent of phosphorylation or the amount of phosphorylation is pretty indistinguishable between patients and healthy controls. But then as we wash out the IL-4 and allow that to go down over time, the patients stay high much longer, suggesting that might help us understand their enhanced biological effect. We also wanted to look at some of the downstream effects of this signaling. And so we've looked at T cell polarization or the T cell phenotyping. And we show in various patients that we can see higher levels of Th2 cells characterized by their IL-4 or IL-13 production through intracellular cytokine staining. We also had transcriptomic data that also emphasized a really big, really large Th2 signal through the transcriptomic signal here in the patients, and more than 50% of those T cells were characterized as Th2 cells compared to just a handful in various healthy controls. And we've also been able to look at other STAT6 targets and look at how they're upregulated in patients versus controls. And one classic STAT6 target is CD23. And this is work done by the Italian group where they looked at their patient and showed much higher upregulation of CD23. They compared it to a number of allergic controls and age-matched healthy controls. So again, I think more evidence that the patient primary cells show an exaggerated response to IL-4, and that's driven by STAT6. So this was nice. We were, working with the patients, and the patient said, it's nice that you can start to understand what's going on, why we have this, but we'd like you to treat us. We'd like to have some therapy so that we can get better, because all the medicines that we've received over the years really haven't been effective beyond the steroids, which have caused us so many side effects. And so we started looking at that. And I think what's nice in this space is that there's a number of potential targets that we could explore that were approved and in the clinical domain. So there's no STAT6 inhibitors that are approved for use in the clinical domain, but there's many medicines that are approved that target this signaling cascade. And so an obvious candidate is dupilumab. This is a monoclonal antibody against the alpha chain of the IL-4 receptor, so important for signaling through IL-4 and IL-13. And then there's an ever-increasing repertoire of JAK inhibitors, so oral medications that target the various JAKs. And so we specifically looked at tofacitinib, which is felt to target predominantly JAKs 1 and 3, and then ruxolitinib, targeting JAK 1 and 2. But there's a range of these medicines. And so first of all, we just went to our cell line data and started to look and see what we could see. So here again, I'm showing you the phospho-STAT6 signal, and either with no medication, in pink with ruxolitinib, green with tofacitinib, and in the blue with dupilumab. And what we saw here in the wild type was that all those medicines, as predicted, were effective at reducing STAT6 phosphorylation. In the patient variants, they were also effective, and something that was quite attractive to me was the fact that the JAK inhibitors were able to really impact this baseline signal that the patients seem to have in this cellular system. So we were encouraged by this preclinical in vitro evidence, and shared this with our collaborative teams, and they were also encouraged. I'll also show you here that tofacitinib was able to bring down that or accelerate the dephosphorylation event that we'd shown was dysfunctional in the patients. Again, suggesting that too might be an effective medicine. And so as we were talking with our colleagues about this, the group in Hong Kong said that their patient had been so sick and they'd been able to access dupilumab because it's approved for severe atopic dermatitis. And this child had been on it now for more than a year and going on 2 years. And that they'd collected some data. And so here's the data for the patient from Hong Kong, looking at here in transcriptomics terms, looking at the signal. So here's his pre-treatment. Here we have a variety of post-dupilumab treatments. And then they ran a panel of healthy controls. And from this transcriptomics, there's a lot of signals, but really maybe the most striking signal is the reduction in these purple cells, the eosinophils. So this may have been the most expensive CBC ever done, where it was powerful data showing in transcriptomic fashion that over time, the eosinophil count fell down. I think it's interesting and matches the clinical experience that sometimes patients on dupilumab got a little worse when they start, and they don't understand why. And then there's some delay and they start to improve. And reading out that, we saw, for example, this very significant reduction in the Th2 cells that they had, and then changes in various molecules that made good sense biologically, including the expression of the receptor targeted by the dupilumab. I think more important than these biomarkers of the response was the patient's dramatic response. And so here, this is again, the Hong Kong patient. You can see his hands before and 2 years after the dupilumab treatment. This is read out through various objective measures of scoring the skin severity with it dropping dramatically. I'm showing you data from this one patient who's been treated the longest, but we have now 3 of the cohort have been treated with a good period of dupilumab and also experienced significant benefit. And this was the most amazing figure of all time, I think. And so we talked about growth being the integrator of health. And so here is the catch-up growth for this young man after dupilumab. So he's fallen off the chart. I've never really seen anyone this far off the growth chart. Here's the 3rd percentile. Here's his height here. They gave him growth hormone to try and have him grow. Nothing happened. And then at age 19, they started dupilumab and he grew by 20 centimeters. He had very delayed bone age as part of his disease, and so had the capacity to grow as a 19-year-old. So he put on 20 centimeters of height and almost doubled his weight. He weighed just over 30 kilos. So what's that, 80 pounds or something, and almost doubled his weight. So really transformative change for him. And then we don't have great documentation of this yet, but some of one of our patients, the patient in Iran, couldn't access dupilumab, but could access tofacitinib. And so the Iranian collaborator wrote to me just last week and said that patient had been on tofacitinib for about 2 months, and they'd gone back and done a biopsy and examined his esophagus, because every month he had to have a endoscopy to balloon dilate his esophagus so he could swallow because of the inflammation. And they went back and it looked much better. The boy had less pain. And then the eosinophil count from the biopsy of his esophagus had dropped down into what was essentially the normal range. So that's, I think, really encouraging that tofacitinib too may be a very effective treatment for these patients. So my last slide would say that I think through this large partnership and by working with others, I think we've started to identify a new inborn error of immunity caused by heterozygous gain-of-function variants in STAT6 that's causing severe allergic disease from birth. We're starting to understand the mechanisms. Clearly, this is early days, but we're showing increased STAT6 activity, increased IL-4 STAT6 axis signaling, and Th2 skewing. I think we've shown both in vitro and in vivo that targeted therapy with dupilumab is effective. And I think also the JAK inhibitors may well be effective. And these are well-tolerated medicines that are administered orally, so that they're very attractive medication. But clearly the full phenotype of these patients will only be understood through the diagnosis of more patients. And really exciting to me actually was a group from Thailand about 2 weeks ago described a family with a father and 2 affected children who also had a STAT6 variant that seemed to be associated with their allergic disease. So I think in my heart, I think this is going to be relatively common in the world of inborn errors of immunity. And I think the challenge for us as clinicians will be understanding when we see a patient with a heterozygous variant in STAT6, which of these are just benign variants and which might be disease causing. think it'll be easy for the ones at 419 because that's a clear hotspot, and maybe relatively easy for others that are enriched in the COSMIC database. But I think there'll really need to be work for us to define these and understand the patients who absolutely have this condition versus those who have just common, more benign variants. So I'll end by saying that this was a huge partnership run by many people. I want to acknowledge the person it's shivering up in Vancouver. It's snowing in Vancouver today, maybe not this much snow. So Mehul Sharma was the PhD student who really led this work with passion and dedication. And then to acknowledge the huge cast of people who generously partnered with us to get this work done. All of them contributed generously to help us understand this condition. So with that, I will end.
I have one question on Zoom, and I encourage everyone who's online to type your questions in there. So the question on the Zoom is, what is the impact of nutritional deficiencies during pregnancy in these patients and on the genetic allergic disease?
Yeah, I think that's, I don't know. It would be the truth. I think these are single gene defects that are fully penetrant, that are presenting in these patients with severe disease. And so I think while all diseases tend to have a gene and environment component when nutrition maybe makes sense as part of the environmental component, I think this powerful genetics is the main driver of what's going on. This condition is a bit different to some more classic, primary immune deficiencies in that these patients are surviving to adulthood, and they're, we saw we're having children. So although significantly affected, it didn't affect their ability to reproduce or to work and function as well.
Maybe I'll start the questioning. So you mentioned a GWAS study, or many, and there was associations with atopic phenotypes, asthma, eczema, et cetera, high IgE levels, eosinophils. Which variants are so common that are picked up on the GWAS study? And were you able to test those variants in your assay so that we can get the link between rare disease and common disease? Yeah,
we've started doing that, but we don't have the data. So that's right. That I really think there's going to be a thermostat of all our genes really, but STAT6 where a little bit up, maybe one of the factors that's contributing to polygenic allergic disease, and then the patients that we've identified here and are describing are all the way on at the, I think, most extreme piece, and I think that's a nice one to look at, which is the variants that sort of turned up time and time again as the variants in the GWAS. The trouble with the GWAS, right, is not designed to identify the causal variants. It's identifying the association. So we've started to look at some of those. Don't have the data. Yeah.
So it's very impressive that you identified the IL-4 STAT6 axis, but have you looked at the IL-13? So usually they're all parallels, right?
Yeah, we haven't. So the truth is, this is sort of very new finding. It's the first time I've talked about it publicly, so I was a little nervous about that. We haven't, we haven't published this yet. It's still as it is unpublished. And so the data I showed you is really what we have in terms of collecting the cohort and defining that. I think there's huge amount of biology that these patients can tell us about these signaling cascades. I think these variants, when we look at them, are, for example, the ones in the DNA binding domain are changing the charge in key aspects of where the transcription factor interacts with DNA. So we have many plans, but also experiments ongoing, looking at ChIP-seq, for example, to understand where does this transcription factor land within the genome now. If are there neomorphic sites? Is it acting as a neomorph or as well as sort of a hypermorph, if we kind of invoke the Muller's morph terminology?
I think we had one more in the chat. If not, then we're either all set.
Oh, yeah. Okay, I'm just doing it with my here. What is meant by rare? Are there genetic diseases that are rare?
Yeah.
GAN, ATM.
Mhm. Yeah.
And then another question is, what is the IL-4 alpha receptor inhibitor or JAK inhibitors used?
Useful. Okay, I can probably. Yeah, I mean, what's rare? I think that's yeah, rare is in the eye of the beholder. At some level, rare diseases are defined as one and less than 1 in 2 to 1 in 10,000 people. I think these are, and then the examples you talk about, sort of ataxia telangiectasia, right? That's a 1 in a million kind of disease. I think my suspicion is that severe allergic disease, and particularly sporadic severe allergic disease, has not been scrutinized and genomically in the same way that we as a community have looked at patients who have severe recurrent life-threatening infections. And so my feeling is that that we'll start to see more and more of these patients with the heterozygous STAT6 variants. That's and I guess time will tell if that's true. You asked about the medicines. So dupilumab, which is the monoclonal antibody against the IL-4 receptor, is used for treating allergic disease. So as you'd expect, so it's been approved for atopic dermatitis, and the sort of list of indications increases. The JAK inhibitors are a group of medicines that, again, the indications have increased. Initially, it was approved for and developed, I guess, for malignancy that was driven by gain of function variants in the JAK, so the myeloproliferative disorders. That's ruxolitinib was approved there. But because they target such a key signaling cascade, indications have increased now. And so the JAK inhibitors are relatively widely used for atopic dermatitis now. Well-tolerated oral medicine. I think they have huge potential. There's even topical versions of those medicines. And so a theme for us as clinical immunologists has been that as we understand and define human single gene defects, we often have now an opportunity to repurpose medicines that have been developed for other examples. And I would say here, we're essentially repurposing a medicine, and that's been showing huge benefit for these patients.
Can I say we have another minute? Can
Yeah.
I ask you another question about
Sure.
the dephosphorylation?
Oh, yeah.
It's unusual that the phosphorylation state, that the baseline's high, the phosphorylated state isn't high compared to controls, but then the dephosphorylation is delayed.
Yeah.
Is that a delay in coming out of the nucleus? A delay in the software?
Yeah.
What do you think is going on there? Just to
Yeah, I'd be lying to say I knew, right? These were the sort of experiments when so my hall as the grad student would get the precious blood came from somewhere. He'd stay up all night doing the experiment. And then he'd be so sad, just no difference, no difference, no difference. And so I think, just thinking and talking to people, we really started to see this dephosphorylation difference. I'd be lying to tell you that I knew the mechanism of why this is happening. I just don't, to be honest. It's new data for us. It seems to be absolutely reproducible in that we're seeing it in other patients. So I believe it's real. But yeah, I don't know why. Sorry. Weak answer.
Maybe use the microphone so the Zoom audience.
do people combine the JAK inhibitors with the dupilumab or the IL-13? in
Yeah,
patients?
yeah. Theoretically, obviously, you can combine things. People start getting nervous about the level of iatrogenic immunosuppression we apply. So, it's relatively unusual to do that. But again, these types of patients who have the signal so exaggerated, makes sense that because they have a germline that's driving it, that with the various medicines, we're just starting to turn it down to zero, but people are nervous. I've shown you the data. We got very good results, it seems, from dupilumab and exciting preliminary results from tofacitinib. So we haven't had to do that. And I guess I should have said, the patients all got off their oral steroids as well when they're on the dupilumab, which is like a huge clinical achievement that made everyone very happy.
Okay, thank
How
you.
we do? Okay. Thank you.